Hello, this is Mrs. Farouk, and I'm going to talk about DNA and the structure of DNA. Um, a monomer of a nucleic acid is called a nucleotide, and a nucleotide has a phosphate group and a sugar, a 5-carbon sugar, and a base. Now, if you remember from our DNA history video, that the famous photo 51 picture of a X-ray crystallography picture done by Rosalind Franklin gave us the biggest clue that DNA is a double helix. And from this picture and measurements, Rosalind Franklin hypothesized that it contained the backbone of phosphate and sugar, and the bases, which she referred as ladder, must be inside of the helix. Now, so when we talk about DNA as a ladder, so here's a ladder, and when we say sugar and phosphate is the backbone, we mean here's the backbones, and then the rungs are like the bases, and it's a double helix because it's twisted. So Watson and Crick took what they know about DNA, Rosalind Franklin's hypothesis that it is a double helix based on the X-ray crystallography. At that time, they note Shugarth's idea that A equal T and C equal G, but they really didn't know what that really meant. And then a nucleotide consists of a sugar, phosphate, and a base. So they decided to make a model, and while they were making a model, they found all those parts coming together. So we have what they refer as the backbone here, the phosphate sugar backbone, which is shaded in blue. And then if you can notice on the other side, when they were putting this model together, that it fit well when the other side was upside down, or what they referred as anti-parallel. So if you notice, the sugar is positioned differently than the other side. And we refer to the anti-parallel by giving numbers to the sugar. So this is carbon 1, and notice you don't see a C because the two lines, when they meet, it's understood it's a carbon there. And this is the second carbon, third carbon, fourth carbon, fifth carbon. And because the fifth carbon is the first one, we refer to this as 5 prime. And then here, the third carbon is the last carbon, so this is 3 prime. And up here, the third carbon is pointing on the top, so we call this end 3 prime and this end 5 prime because the fifth carbon is pointing there. And then, as you can see, in the rungs of the ladders are nitrogen bases, where A will pair up with T and C would pair up with G. So there are 4 bases, and the bases are classified according to the ring. So these are pyrimidines, cytosine, thymine. Now, uracil is not found in DNA, but it is found in RNA instead of T. And then you have purines with 2 rings, adenine and guanine. And notice here the 5-carbon sugar, RNA has a ribose sugar. Notice that oxygen right there, where DNA does not have that oxygen, so that's why they call it deoxyribose. And then here's a nucleotide. So both RNA and DNA have a phosphate group, 5-carbon sugar, which they could differ, and a nitrogen base, which both have cytosine, guanine, and adenine, but RNA has uracil instead of thymine. Another nucleotide that we know of is ATP, and ATP is made up of ribose sugar, 3 phosphate groups, and an adenine base, so that's why it's called adenosine triphosphate. And as we know, when we break that bond by hydrolysis, we release energy and we have ADP. And when we store energy back, then we add that phosphate group and then we have ATP. And we can even further break down ADP into AMP and release even more energy. Now, nucleotides again are monomers of nucleic acid, and when you want to add a nucleotide, it's done by a condensation reaction. If you remember, condensation reaction is when you remove a water molecule. So here, as you notice, here's the 3 prime, the third carbon. This hydroxyl group forms a bond with this phosphate, and that's how you would add a nucleotide. Okay, and this is going to be really important now when we talk about replication. So here's a closer look of the condensation reaction forming that phosphodiester bond, and this bond is really strong. If you think about it, we found DNA in fossils. We were able to recover, and it's more heat tolerant than proteins are, and it's due to this strong bond between the phosphate and the sugar in DNA. So DNA is a double helix, and it's arranged in an antiparallel fashion, where RNA is a single helix. It doesn't have that other backbone. Okay. So summarizing about nucleic acid, key elements of DNA has carbon, hydrogen, oxygen, nitrogen, and phosphate. Its monomer, remember, is a nucleotide, has a phosphate group, a sugar, and a nitrogen base. Its polymer of nucleic acids called DNA or RNA. And key bonds to know, the hydrogen bases are between the bases, and the phosphodiester bond is between the sugar and the base. And key function is, of course, DNA is the hereditary material, regulates cell activities, and an example, ATP is used for energy. So I'll conclude that, and thank you for listening.